The overall goal of this experimental procedure is to demonstrate the fabrication process in subdiffraction imaging of the two-dimensional hyperlens device. This novel super-resolution imaging technique has the advantages of real-time imaging and simple implementation to conventional optics.
This method can help answer key questions in the field of super-resolution imaging, such as imaging living cell and dynamic nanoparticle below diffraction limit.
The hyperlens is a special spherical lens with multi-layer structure, having a flat hyperbolic dispersion that supports magnification of high-frequency information and resolution of small objects in the far field in real time.
The main advantage of the spherical hyperlens is that it can magnify two-dimensional information at visible frequencies. A spherical hyperlens can also be easily integrated to the conventional microscopy without additional complex system.
Demonstrating the procedure will be Dasol Lee and Inki Kim, who are graduate students of my laboratory.
To begin, spin coat the quartz wafer with a positive photoresist at 2000 rpm and bake for 60 seconds at 90 degrees Celsius. Then, use a dicing machine to cut the wafer with photoresist into small pieces 20 by 20 square millimeters in size. Blow the pieces using a compressed nitrogen gun to remove any particulates resulting from the cutting step. Next, place the cut wafer into an ultrasonic bath of deionized water for 5 minutes at 45 degrees Celsius. Remove the photoresist layer using an ultrasonic bath of acetone.